On behalf of DG, our director, and ICGEB fraternity, ICGEB whole fraternity, I welcome you all. And it has, and also we thank you for making this, coming to this and having a beginning for our ICGEB alumni meet. It has been a great pleasure for all of us to have you again. And as you know, last 35, ICGEB is about to complete 35 years, and we have many students who have really reached to the very higher levels, and they have done really remarkably well. And with all of us thought that we should have you with us again, and we also will now next meeting will also will need your requirement. We want to involve you with us to organize many such more meetings. So with these words, we welcome you again, and I invite Dinkar to say a few words.
Well, good morning to all of you, the ICGEB alumni and the ICGEB future alumni. So, this is a great event, and it is wonderful that we have such a strong response from alumni that I can see emotional link that they have, having worked in this institute for several years in the past, even though in some cases it could be 20 years, 30 years. We, I think, have alumni here, both in whole of ICGEB, first alumnus is present here, and in ICGEB New Delhi, first alumnus, first graduate from New Delhi also is present here. That's a wonderful thing to find. The idea of having these meetings began in 2019, and we earnestly started planning for it to have a meeting in 2020, and we approached quite a large number of friends who have worked in the past in ICGEB. In fact, Pawan Malhotra and Dheeraj Kumar did remarkable work in getting together with substantial cooperation from Suzanne in the headquarters. But unfortunately, pandemic really messed it all up for us. But then I think we restarted this thinking just a short time ago, and in spite of a very short notice, many of you responded so well, and I'm very glad both DG Lawrence Banks and Susan is also here, and whatever you see the way it has got together is real efforts of Dheeraj and Pawan, and I must credit competent support from Sweta as well as my office team. So I think it is the team effort among these people that really brought this program together, and no less credit to our alumni who are here, and we are going to listen to them, we are going to interact with them, and as you all know, we will have the annual lunch of ICGEB preponed just to coincide with this. So is the student symposium. So we have packed it in such a way that actually all the staff, present staff as well as students will get to meet alumni in this event, as well as alumni will get to learn little more about what present research is going on in ICGEB in the afternoon session. So this is really an event that has been put together remarkably well, I believe, and let us enjoy it. And I again thank all the alumni who are present here physically, as well as many of them are online. So let us enjoy the day which is essentially meant for the alumni. Thank you.
Thanks, Dinkar. Almost all the ICGEB faculty has been involved in organizing this event, and we should not not forget the name of Dinesh, who has really set up a very good website, and so that if we have forgotten some of the names, please tell your friends that we regret that, and you can add some of those names, so that we still don't have a complete list. We would like you to please involve other of your friends in this whole alumni, ICGEB alumni. So now I invite our DG to say a few words, and
thank you, Pawan. Really, I'd just like to reiterate what Dinika said. A massive thank you to the whole team here in New Delhi who have really made this possible. So, good morning. Welcome back to ICGEB. It really is a joy to see so many old friends and colleagues all gathered together in the same place after so many years. So really, and thank you so much all of you for making the effort to be here. You really do represent the whole of ICGEB's history, from its initiation of activities 35 years ago, really up to the present day. And I think you're a testimony to ICGEB fulfilling its mandate. We're a centre of excellence for providing education and training in molecular biology and biotechnology. And I think we really bask in your success. You've gone on to be world leaders in your respective fields of endeavour, and that is something that we're really gratified by. Indeed, we do strive to provide an environment which can nurture such growth and thereby cascade our impact through your successors. You have all gone on to be, as I say, world leaders. You've gone back to disparate parts of the world and really, really making a major impact back where you are. And that's exactly what we are founded to achieve. And I think your individual brilliance in many cases has really grown greater than your mentors. And that's exactly what it should be. We should all strive to ensure that the next generation can do even better and have more of an impact for mankind than we've been able to achieve. That's the whole thing, improvement. And so again, congratulations to all of you who have done so well. So thank you again for being part of the ICGEB Alumni Association. This is the first such gathering that we've had, and this is not going to be the last. We really have a strong intention now to make this a regular event. And I feel that this network is amazing, and we really want to capitalise on that, both from offering opportunities for you to re-establish old contacts and also make new interactions and new collaborations, which hopefully will be of benefit both to your own science, but also for furthering our collective mission of making science, basically making the world a better place. So thank you again for coming. It really is tremendous to see you all. I'm really looking forward to a fantastic day. Thank you very much.
thank you, Lawrence. Now we begin the first speaker. I know most of you know him. He is the inventor of first mRNA vaccine in India, and I think this is going to be in the distribution next month. Already. Okay. So Sanjay Singh, most of the speaker, I think they will introduce themselves about which lab they belong and all those. But Sanjay Singh was in malaria group, And in fact, I still remember the day when I and Chetan went to interview him in Lucknow for a postdoc position. And he was always enthusiastic to work in ICGEB. And he has that imagination that he wants to reach to some heights. So we could see that sharpness in him. And here he is. He is the CEO of Genova, and has really done remarkable work in development of vaccine, and has also been able to give vaccine in the fields. So I now invite Sanjay to present the first talk among the alumni.
This is not the first slide. Yeah. Open it.
So, thank you, Professor Lawrence Bank, thank you, Dr. Alumni, thank you, Dr. Malhotra, and thank you, Dilip, for inviting me here, and it's always a pleasure to come back. ICGEB is one of the important milestones in my life, because I come from a family which was half medical side and half was the physics. And when I decided to do biology, everybody was quite stunned, especially my father, because he will not able to understand logic if you are not able to do physics. But from day one, my interest was in applied side of the science. And after completing my and we feel very proud of it. Now I'm going to change the gear. So this is all going on. So then we move for the same drug for next indication. And here, being a scientist, you have to learn something which I was not knowing, Professor Lawrence, that science can come into these parts. So stroke is even worse than heart attack because here the neurons goes down and finally it is the biggest CD4 positive interferon gamma positive T cells. This is also in many logs fold. So this is the one thing which has made a huge difference into us. And at present, we again went for a trial, and this is I'm really being a scientist, that how the system sometimes is biased. We have done 1,000 times more sample what has been done for my competitor vaccine. But when we went for this, they should say go for 3,000 more. We have completed 3,000 more. 140 we did intramuscular, and OM is the intradermal, Omicron is intradermal. We completed this, and then we have gone for this 3,114. And Malik, yesterday I was not able to take full because just yesterday afternoon we completed this 3,142 volunteers. These are very big trials. They cost hundreds of crores, and 22 centers are going on 24/7, because once you immunize people, you have to watch them for a week, for 24 days, and there's no difference coming. I know somebody is having headache, they say because the vaccine has failed, but you have to send the doctor, they have to analyze, you have to bring back. It's a huge task, but we are very happy what we are doing. So this has gone through, and why we are very excited about it, because we feel this is disease agnostic platform for vaccine. Disease agnostic, why? Because these all components like nanolipids, enzymes, cofactors, all are common for anything. Only the difference is you put the plasmid, you change the sequence, and you can have the vaccine available. And this is disease agnostic. Any disease it can go, any variant it can go. So we are working on certain more diseases, and the stockpile can be kept anywhere. So this is, as I said to you, and here we have broken the technology into 4 parts. One is plasmid, second was mRNA, third is CRNA, and fourth is the RNA vaccine. And it can be produced at different places in the world. And so we are what is going today, we have divided the science into 3 parts: mRNA part, backward integration part, and disease biology part, because here we go and collaborate with research labs for B-cell, T-cell biology, or optimum antigen target, or protection. And this side we have made the first and the second is going, and we feel that there may be some addition is required for the real. Actually, we do not know, because as a whole scientific community, we have not able to make a flu vaccine universal in the last 40 years, but still we have the hope we can do something. And then we are working on singles, dengue, and malaria kind of, and then novel vaccine, tuberculosis response, these kind of things. This is the last slide we want to present. So I was invited by the WHO, because WHO decided to make mRNA hub all over the world. Why I have kept this slide? This was the motto of ICGEB very early in life. We can take our biotechnology to the next places. And now is going to help around 39 countries to work on this mRNA as one of the leads. So I think this is my last slide. So thank you very much.
That I do agree with that.
Thank you, sir. Thank you very much.
He has been star earlier also, and he is still a star. So we will take few questions because of time short. Any has sure.
Last part of your talk on replicating version of RNA vaccine. It seems that whatever platform you use, durability and how long antibody persists is a major issue with whatever technology you're using. So are there any data to sort of compare whether the durability of antibody response generated is long lasting with these platforms?
Yeah. So Sanjay, this was really great reminder of, you know, how things can happen so quickly, and and you reminded us of this samples going into the bloodstream, and you know when we were kids, every immunization, you know, they will pull back, and as you said, nobody does it now. And there have been a lot of reports that not only in the heart, the mRNA can even go to the liver and kidney, and you know, and a lot of people did not believe that data. They said, no, these are some artifacts, because messenger RNA should, you know, completely disappear. But you know, the kind of data you presented, that I think it's really important that the vaccine should not get delivered into the bloodstream, not only for the heart protection, but for the liver and for the kidney, for many organs. So really enjoyed your presentation, great going, and God bless you.
Thank you, sir.
Yeah, nice to see you again, Sanjay, and it was a really very stimulating talk. I hope everyone enjoyed. So I have a small question that the ECG based solution which you showed us, is it similar to what Apple is already giving in market, because it does not work on our Android phones, and most of the poor people have Android compatible phones only.
No, so it is not on the phone.
Okay.
I will not go on, I don't wear a watch.
Okay, so it's a different instrument.
So this is a whole set of instrument which is attached to 3G or 4G.
Mm-hmm.
So this is proper ECG, 12 channel
Lead,
ECG.
okay.
12 channel ECG. So as soon as the patient has come to the hospital, we have taken the 12 channel ECG, and this ECG machine will automatically write, because it has around 5 million cases data to artificial intelligence built in.
Mm-hmm.
But it will also fly back to the headquarters, the command office, and there also it is analyzed, and based on that the signal is given, yes, this is correct, go ahead. So this is the mission Delhi Command Hospital has been made at AIMS with Professor Balram Madhav, and we are the drug part, because we were having this dream. I came to talk to him a couple of years back, then ICGEB funded this program, because government said government want to test by its own that what we are saying. So now they have the data, and hopefully you might be knowing a term, aspirational district of the Prime Minister, 145 districts. So hopefully it is in process of implementation. So we will save hundreds of thousands of lives every year, which through hardcore biotechnology, and where ICGEB played a very important role in my life. Thank you.
Thank you, Sanjay.
Thank you, Sanjay. I now request Professor Dinkar to present a moment.
Thank you, sir. And I would like to say another relationship which is known to public. When I was working ICGEB, Dr. Salunke's lab, my wife was working. She was trying to learn some protein chemistry and crystallography. She was from chemistry. And so every day I go to him in the evening and see, but he's a hard taskmaster, so he's sometimes not finished till 9 o'clock. So I sit down into his room and talk many things. So I have a long relationship, I think it is now 25 years back, long relationship where, and he is still looking as young as he was.
I have protected support system of his success.
Thank you, sir.
Thank you, Sanjay. It was a very informative talk. Now we move on to our second speaker, Stefano, who has really worked hard to reach this place. I must tell you that we booked tickets and then it got cancelled, and now he actually came on his own to this place, and really from Italy, and we really thank him for all this. And I must tell you, yesterday we came to know that he's first alumni student, PhD student from ICGEB, trusted. So let's give him applause, and he will, although he will introduce himself, but he will be talking on non-coding RNA and their use.
Thank you.
and biotechnology, and biotechnology. Actually, at that time, we're talking about too many years ago, the best place in Italy was Pavia, okay? And Falaschi's lab was famous at that time, was really the leader in the field of molecular biology biotechnology at the CNR, Buzzato Traverso. And so I decided to apply there, and actually I got a PhD position there. But then, I wanted to move from Trieste to Pavia. I am from Trieste. But then the labs in Pavia moved to Trieste. So it was somehow a strange situation. So I decided, okay, I don't go to Pavia, I stay in Trieste, and I applied to ICGEB. Actually, I applied to this thing that was starting at that time in 1988, when these two, incredibly great scientists and intellectuals, Professor Falaschi and Professor Daniele Amati, that was the director of SISAM, International School of Advanced Studies in Trieste, decided to start the first joint PhD program, ICGEB and SISAM. And actually, it was a program in molecular genetics and biotechnology. And as correctly was said, I was the student, we were in three actually. I was with Francesca De Marchi from Falaschi's lab, and actually we were the first one to get the PhD, to start it and to get the PhD. And I still remember the fantastic courses they organized in the first year. So I'm really, really grateful to ICGEB for coming to Trieste actually and setting up this fantastic PhD program. Today I'm going to present some data from my lab about a specific new functional class of non-coding RNA. Since the sequencing of the human genome, we learn two interesting things about the general organization of a mammalian genome. On one hand, that in addition to 25,000 protein coding genes in the mouse and human genomes, we have actually 35,000, so 35,000 genes that are transcribed, but they are not translated. They give rise to the so-called long non-coding RNAs. Actually, we now know that there are many more long non-coding RNA genes than protein coding ones. On the other hand, we also learn, surprisingly, that basically every single gene in almost every genome has another gene, at least another gene in the other filament. So they form what is called a sense-antisense pair, and this is important for many points of view. Okay, just think about it. When you do a knockout of a gene, you are also knocking out a gene on the other filament. When you have a mutation, you have a mutation also on the other filament. So really this complexity of the transcriptional units in the sense-antisense side actually really have a lot of functional consequences. So if we look at how a sense-antisense pair can be organized in a specific genomic locus, we come out with three different possibilities. One gene is completely contained in the other one. A second one, two genes are actually in convergent transcription and they overlap at the 3' end, or they are divergently transcribed and they overlap at the 5' end. And you can imagine all the different possible combinations. You can have a protein coding gene that is antisense to a protein coding gene, a long non-coding RNA that is antisense to a protein coding gene, and a long non-coding RNA that is antisense to a long non-coding RNA. Actually, the large majority of studies have been focused on how a long non-coding antisense RNA can regulate expression of a protein coding gene on the other filament. We were very curious about this, and actually we studied we isolated and studied long non-coding RNAs that were antisense to genes involved in neurodegenerative diseases, specifically in those genes that are mutated in the hereditary forms of Alzheimer's, Parkinson, ALS, and Huntington. And I want to, the complexity is staggering. We discovered 7 new long non-coding RNA transcript of a gene alpha-synuclein. That is a gene that has been incredibly studied because for its importance in Parkinson's disease. But this complexity was missed. On the other hand, we concentrated our attention, we focused our attention on a specific sense-antisense pair at the gene encoding for UCLH1, a gene involved in Parkinson's disease and Alzheimer's disease, that is down which protein product is involved in ubiquitination, and it is a component of Lewy bodies, that is the neuron pathological hallmark of Parkinson's disease. Let me describe this, how this sense-antisense is organized, because it's central to my talk of today. So on one filament, we have a protein coding gene, that is UCLH1, that is transcribed, it has an AUG and stop codon, and gives rise to a UCLH1 protein. On the other filament, in a head-to-head divergent conformation, so in a situation in which the two genes overlap at the 5' end, we have a long non-coding RNA. This is the final transcript, this is the gene with internal exon, and this is the transcript. As you can see here, you have a regions of overlap between the two transcripts, that is around the AUG, and that is at the 5' end. And this overlapping region is only of 73 nucleotides. But then you have also a non-overlapping regions that in the case of the antisense to UCLH1, so the long non-coding RNA, contains an embedded transposable element and embedded repeats. That is of two types, is a sin B2 and is in a halo. So these are the two crucial characteristics of antisense UCLH1. So then we cloned the full length of antisense UCLH1, we transferred antisense UCLH1 to cells with dopaminergic neurons that are expressive of the sense of the antisense, and we got a surprising result. Actually, by transfecting the long non-coding RNA that is antisense to the protein coding genes, we got an increase of UCLH1 protein level. And this was occurring at post-transcriptional step because there was no change in UCLH1 mRNA level. This clearly was surprising because the first bet that you can do, about an antisense sequence is that it's working as an interference. So it's downregulating. Actually, it was happening quite the opposite. So then, we made a lot of, we treated the long non-coding RNA as a protein coding gene. So what you do a mutational analysis, deletion, you create new constructs, and we understood two things. One thing that it was crucial, that the ability of the antisense to UCLH1 to increase the UCLH1 protein level was dependent on two RNA domain sequences, okay? A long non-coding RNA has domains, not made by amino acids, but made by RNA. And these two domains were clearly the antisense overlapping region, okay? And one of the repeats, particularly the sin B2 element. Sin means a short interspecies nuclear element. There are 350,000 in the genome, and one of them actually was embedded in the transcript in an inverted conformation and was fundamental for the ability of the antisense to increase UCLH1 protein level. Since we published this some years ago now, actually I don't want to go into the details because these are all published, but I just want to give you what is the biological function of antisense UCLH1. Okay, so basically what's happening in physiological condition, antisense to UCLH1 is in the nucleus, and UCLH1 is translated in the cytoplasm, being a messenger RNA, it is translated. However, when stress condition occurs, therefore it means that we have an attenuation of cap-dependent translation, the antisense to UCLH1 shuttle from the nucleus to the cytoplasm, form the pairing, binds the ribosome, and increase translation of UCLH1 mRNA. Okay? So let me repeat, antisense UCLH1 to the binding to UCLH1 mRNA, load UCLH1 mRNA to the ribosome, and increase its translation. So this explains why there is no change in UCLH1 mRNA level, but an increase in UCLH1 protein level. And this is happening in stress conditions. But then we found out that actually there are more than 200 sense-antisense pair with a protein coding gene with filament, a long non-coding RNA with a filament in a head-to-head divergent transcription with an inverted sin B2 element, actually that have the same characteristics. We tested more than 10 of them, proving that they work exactly the same, in the same way, and so we found that this could be a new functional class of long non-coding RNA. And we call them synapse, because to the activity of a sign element, they are able to upregulate translation. Actually, when we confirm it with a different type of embedded transposable elements embedded in London codon, there are more than 1,000 in the human genome, okay? And the mechanism is conserved even in Drosophila. So it's really a general mechanism of regulation of gene expression. Okay, but we thought, okay, this construct is able to increase translation of USLH1. Clearly, it depends on the binding domain, because it's binding the mRNA for USLH1. What about if we substitute the binding domain to USLH1 with a binding domain to a gene of interest? And actually, we did that as a proof of principle by creating an artificial sign-up against GFP, okay? So that to show if we are able to increase GFP protein 3 synthesis, okay, by simply, putting a specific binding domain. And actually, this is what is happening. It is actually a sign-up against GFP is able to increase GFP protein level with no change in USLH1 mRNA. And from now on, we basically say, okay, the synapse and the inverted SMB2 elements is an effector domain when the biological function takes place, and the binding domain is actually specificity, confers specificity. So to what mRNA I'm going to increase translation, which mRNA I'm going to increase translation, this is driven by the sequences of the binding domain. But then, since the binding domain and the effector domain, so the antisense sequence and the inverted SMB2 are required, then we also show that they are sufficient. So now you can have a mini sign-up, because antisense USLH1 is 1,800 nucleotides long, but if we put together the binding domain and the effector domain, we have a 250 long-non-coded RNA long. So it's much shorter, and you just need the effector plus the binding to increase the target protein levels. But then you ask yourself, am I going to be able to create a synthetic sign-up direct against an endogenous gene that normally does not contain a natural sign-up in the genome in a sense-antisense pair? And this is an example in which we design a sign-up against DJ1, a gene involved in Parkinson's disease, and we were able indeed to increase endogenous DJ1 protein level with no change in DJ1 mRNA. Now, 3 questions. What is the molecular mechanism? Second, how many of them do exist? Can sign-up be a new platform for RNA therapeutics? Since, we can increase endogenous protein synthesis of a gene that we don't. So let's go for the first question. So first of all, we did chemical footprinting, NMR, in order to understand the structure of the inverted SMB2 element, where the biological activity is contained. So the inverted SMB2 element has formed these sequences. These are 5' end, goes up and then goes down. This is the 3' end. There are 4 internal loops, 3 stem loops. Stem loop 1 at the very top is indeed fundamental for function, because if you delete only this part, there is no activity. You lose the activity. We made then NMR of the stem loop sequences. It forms an A-type RNA helix. But what is important, it has a pseudo loop at the top with a GUG3 loop, a 2 highly unstable AU base pair. Okay, since my PPT was not formatted well, I have here a PDF, so I cannot show you a molecular dynamics simulation movie of a NMR data of a stem loop 1, but you can see here these are the 3 nucleotides that just are at the top of a structure, GUG. We will see later it's fundamental for function. But you remember it was shuttling from the nucleus to the cytoplasm in stress condition when cap-dependent translation is attenuated. And we know that in those conditions, internal ribosomal entry sites are important. So sequences that give rise to specific secondary structures that bind directly the ribosome and get translation. This is important because the machinery of translation, it's up 40% of intercellular energy. So when stress condition occurs, we need really to translate everything that is related to surviving. Okay, so we ask ourselves, is since internal ribosome entry sites are fundamental for translation in stress condition, could it be that the inverted SMB2 element, a transposable element in the London coding RNA, is indeed an internal ribosome entry site? And first of all, you need to show that actually is binding the ribosome. And this is polysome profiling, in which you can see here, this is free RNA, these are 40S, 68S, and then there is light polysome and heavy polysome. And you can see here that the anticellular H1, the synap RNA, is actually associated mainly to 40S, and is not associated to polysome when translation occurs. On the other hand, UCLH1 mRNA actually is strongly associated in low polysome, in light polysome and heavy polysome. We will see later how synap works. So this was, encouraging. It is associated to 40S, where normally iris sequences associate. So but there is a bicistronic assay to prove that the sequence is an iris. Basically, you make one single mRNA, and you have two operating frames. So AUG, stop codon, a sequence, AUG, stop codon. If you don't have internal ribosome entry site here, only the protein first position can be translated. But if you have an internal ribosome entry site, you can also here see a translation of the second position operating frame. And so we tested the inverted SMB2 element from anticellular UCLH1. The SMB2 element in direct conformation, because it does not work, and the positive control, that is CMYC iris from CMYC. And as you can see here, in this assay, actually the inverted SMB2 element was having more iris activity than the positive control. And this activity was completely killed when it was direct. So creating a relationship between synap activity and iris activity of inverted SMB2 element. But then, look at this. So this is an experiment with transfect synap DJ1, again DJ1, we do polysome profiling, again, synap DJ1 associate to 40S, okay? And actually, associate, the control here is the synap without binding domain, so it must not work. But, interestingly, the mini synap without binding domain associate anyway to the polysome, okay? To the 40S, suggesting that the inverted SMB2 element itself is binding. On the other hand, you see, this is the distribution of DJ1 mRNA in the control cells, but this is the distribution upon transfection of DJ1, of synap DJ1. And we have several examples of this type of distribution, where the association to heavy polysome of a mRNA target is increased by transfection with synap DJ1. So indeed, we are increasing this association. And it's interesting to understand how, since synap does not go into the polysome, remains in the 40S. Then, what we could, but at the meantime, something new happened in the field of translation, because a new player gets into the town, that is M6A dependent translation. What is M6A? Basically is a epitranscriptomic modification of adenosine, okay, here with a methyl group, and this is a signaling as a signaling function within mRNA, and and the pathway is heavily regulated by writers, that particularly methyl 3 that methylate the RNA, by erasers that take it out, and by readers that note the presence of a methylated adenosine and trigger a biological response. So we were wondering, is it are synapse M6A methylated? And this is part of its activity as translational activator. So first of all, we proved by mRNA immunoprecipitation with an antibody against M6A, the synapses are indeed M6A methylated. And actually this signal disappears if you do the same experiment in cells that are knocked down for methyl 3, so the major methyl transfer is in the cell. But what was more important here, that actually if you do if you transfer synap DJ1, okay, in cells that do not express methyl 3, so you don't have methylated synap, actually you not only lose the activity, but it acts as a dominant negative activity on endogenous DJ1 protein level. This was incredibly surprising. We have no idea why. Okay, so we decided to focus our attention in solving this apparent contradiction. So we resorted to nanopore. Nanopore sequences is a fantastic technology that we have set in the lab. We already sequenced more than 100 human genome with a nanopore technology. And by nanopore, you can actually look at the post-transcriptional modification in real RNAs. And we found out by using nanopore that we identified the two adenosines that are modified in synapse RNA. One is A46 and one is A110. Okay? So we validated these experiments. I don't want to go into the details. We made mutants. We mutated both adenosines that are supposed to be the site of MC6A methylation. And we prove it, by immunoprecipitation. And again, we found out that the double mutant was a dominant negative of endogenous DJ1 protein level. So we confirmed this pattern. And then we resorted to ribosome profiling again, polysome profiling again. So this is, the control situation, which you transfect DJ1, the synapse DJ1 without binding domain is associated to 40S. This is the distribution of DJ1 mRNA. This is what's happening when you transfect the mini synapse DJ1. Again, is here, it is here. Look what's happening here. When you have really an increased association of DJ1 mRNA to polysome as expected, and this leads to an increased endogenous protein level. But when you transfect the double mutants, again, you have a synapse DJ1 associated to 40S, but basically what's happening, you decrease the quantity of DJ1 mRNA associated to polysome. And in doing so, actually, you accumulate endogenous DJ1 mRNA to 40S in an inactive manner. So it's really acting dominant negative, sequestering the bound DJ1 mRNA into 40S and not allowing it to enter into active translation. So right now, we have this model for activity. MC6A methylation of synapse RNA occurs in the nucleus because method 3 is a nuclear protein, and then we have a formation of sense-antisense pair in the nucleus. So this is the binding domain, this is the SINB2 effector domain, and these are the two methylation sites. Okay, they bind to the messenger RNA, then they shuttle to the cytoplasm, where the effector domain acts as an internal ribosomal entry site in trans to the binding domain. Okay? We now know, I don't have time to show you, that the mechanism, that is the UG that interacts physically with forming nucleotide pairing with 18S of a ribosome. And then something happens that is M66 dependent. So the synapse has to so first of all, the pairing must be unwound because the AUG is covered here by the binding domain, okay? And not only this must be unwound, but the synapse has to be detached from the RNA. And the mRNA enter into the tunnel and becomes to be translated upon formation of ATS ribosome through the addition of a 60S, okay? And on the other hand, the synapse becomes solitary and detached. We are now studying if it is degraded or if it is reutilized. But let's go to see the complete description of synapse. Because we have just shown that the inverted SINB2 element is an iris that acts in trans. Is it a general characterization of irises? Could it be that the irises, in addition to work as in cis, also works in trans? So we took the iris of CMYC, that was our positive control, and we substituted the effector domain of inverted SINB2 element, put in the CMYC of iris. And actually it works. It works exactly as the inverted SINB2 element. Look at here. So this is the synapse control with an inverted SINB2 element as an effector domain. This is a synapse in which CMYC iris is an effector domain. And is able to increase protein levels in trans. So then we tested 8 different irises, 4 from viral origins and 4 for cellular origin, and all of them works, actually with different types of activity. Right? This is synapse is 1 levels, but all of them works as able to increase in a post-transcriptional manner translation of a target in trans. So these are synthetic constructs. Do indeed, are indeed in our transcriptomes RNA, natural RNA, that works with an iris in trans. So we look at that, and we resorted and we focused our attention on circular RNA. Because circular RNA is known that they contain irises, and those irises are active because they are able to drive expression of an operating frame that is in the circular RNA. So what we did, first of all, we proved that the synapse GFP is active in a circularized RNA conformation. And then we decided to study a specific, we took 2 databases, the database of iris sequences and the database of circular RNA. We put that together and we looked for circular RNA containing iris sequences with no operating frame, because it's a non-coding circular RNA. And we focused our attention on circular 5533 that is done exactly like that. It is a transcribed for the CMYC 5' end. It contains the iris sequence, no operating frame. We transfected it, and we did mass spectrometry and RNA-seq to look for proteins which expression is increased with no change in mRNA level. So we focused our attention on one target, that is PXK. Actually, PXK protein at post-transcriptional level is increased when you transfect circular 5533. And then we look bioinformatics for binding domains, because these are not, in a sense-antisense pair. Circular 553 is synthesized in one locus, the target is in another locus. So we have now to find out how they bind. And we found different target binding domain and target sites. We did all the different types of mutational analysis, and I just give you the take-home message. Basically, the binding occurs in this way. We have 2 binding sites in the circular, in the non-iris sequences. One is targeting a sequence at the 5' end of the mRNA, and one is targeting the sequence at the 3' end of the mRNA. So the situation is different. We have 2 binding sites, 2 target sites, one 5', one 3'. And this is actually what's happening at the polysome level. So circular 5533 is associated to the polysome, not only to 40S, also in the polysome, and we don't know, we have now to understand how this is possible. But again, when you transfect circular 5533, you get an increase of PXK mRNA association to the polysome, increasing a protein translation. So let me get my last 5 minutes of my talk. I don't know what is the time, if I'm late. Okay, 5 minutes, 2 minutes. So I just want to give you the examples of how you can use synapse technology in therapeutics. Okay, why synapse could be a new platform? First, it acts on specific mRNA to the binding. It increases in physiological level from 1.5 to 3, so you don't get an overexpression of tenfold, but sometimes is important. And you have no uncontrolled ectopic expression. No expression of mRNA, no synapse activity, more specificity. So you could guess 2 types of delivery, through an AAV or with an azosynapse as an oligonucleotide. I don't want to go into detail because I don't have time, but at the end we were able to get down to the sequence of a synapse to less than 40 nucleotides. Because we actually using the stem loop that is at the top of the structure as an effector domain. And this was happening for this nanosynapse that was chemically modified with 2' modification, in which we have the stem loop 1 as effector domain, the binding site less than 40, this is 53, but we are now less than 40 nucleotides, and it works exactly as the synapse delivered to a plasmid. 2 fast applications, one directly target genetic lesion, compensate for disease condition. Directly target haploinsufficiency. All of you knows what haploinsufficiencies are, is when you have a genetic lesion that delete one of the 2 alleles, so you have 50% of mRNA, 50% of proteins. There are more than 300 diseases, all untreatable. Okay, so synapse is perfect for this, because you have 50% of RNA, but you increase translation, you double the protein level. So it's in potentially therapeutics. So we got a proof of concept with on Friedreich ataxia, where frataxin is less than 50%. This is just to show you the level of frataxin protein and the decrease in mitochondrial activity in Friedreich ataxia, because frataxin is a mitochondrial protein. Here you can see that by transfecting synapse against frataxin, we restore the physiological level of endogenous protein level from patient-derived cells, and we restore also mitochondrial activity. But most importantly, in vivo, GDNF, glial cell line derived neurotrophic factor, is a factor that is considered to be therapeutic for Parkinson's disease, but many clinical trials fail because, if you overexpress too much, it becomes toxic, so on and so what. So we designed in the synapse for GDNF. This was the activity in vitro, increases the secreted level of GDNF proteins. Then we did, we produced an AAV that delivered the synapse. We injected on the striatum. This is the level of endogenous GDNF proteins after 6 months from injection, in the striatum. So you get almost threefold increase in endogenous GDNF production. Okay, and this was having many effects as we were expecting. For instance, increasing dopamine release upon amphetamine of potassium chloride injection. This is in living animals with some devices in the brain, implanted in the brain, or in a neurochemical model of a disease that is a classic 6-dopamine neurotoxical model. Okay, actually the co-injection of synapse or AAV deriving synapse expression was reducing the behavioral alteration due to loss of dopaminergic neurons, and was reducing the loss consistently, the loss of striatal terminal, dopaminergic terminal in the striatum of dopaminergic cell number. So really, we show you that indeed synapse GDNF could be active in vivo, and the protein that is produced with the synapse mechanism is functional and has therapeutic potential. Right now, what we are doing multi-synapse. So we are putting more binding domain in the same synapse. And the first example is here, in which we not only make a synapse against GDNF, but also against the receptor. So in order to increase the entire neuroprotective pathway. And here, just to give an example of a multi-synapse in which you have a 5' end, the binding domain for RET, the receptor of GDNF, and then the binding domain for GDNF, and then the inverted M2 element. As you can see here, we are able to increase twofold both the receptor and the protein. And right now, we are doing synapse successfully with 4 binding domains. Okay? And I just want to say that this is all IP protected, this led to establishment of a company in Cambridge, UK, Transign Therapeutics, got 15 million as money, and we recruited an industry veteran, Jan Ticklet, as CEO last year. And I'm very happy to take your question. Thanks a lot.
All the speakers will be there during tea time as well as lunch time. We can restrict this, and I also request next speaker for 20 minutes time. Otherwise, we'll not have a limited. We have a limited time. Will not be able to come. Now I request the Dinkar to Stephen. Next speaker is Shweta. She has been star at ICGEB when she was doing PhD, and she has again done very well in her own lab at CDFT. So I'll request Shweta just to restrict to 20 minutes if possible. And she has been continuing on hepatitis E, or no? Okay.
We'll just see in a minute.
Okay.
So I would have a mic, right? the speakers should stick to 20 minutes because, otherwise we'll never finish and our memories are kind of will go on and on. So as you would see, I have changed the title of my talk, and when I gave you the title, I had thought that I would put in pictures and I would talk about, when I was last here, and then I realized I moved so many times that I don't have access to the pictures, and I went to my parents' place yesterday to find those pictures, and they pointed me to a, top shelf in the loft, and I decided it's easier to change the title. So that's what I'm going to be talking about, how the histone little transperases regulate diverse cellular processes. So, but I do have this slide. Okay, so this is kind of just giving, what I have done for the past few years from my PhD in biology under Dr. Sunil Lal, and also equally contributed by Dr. Jameel and Dr. Vijay Kumar, because of course we were a group. And then I did not go for my postdoc immediately to the, my bosses were very upset with me, but I got married during my PhD, and then I spent a year in Bangalore in a startup called Vistagen. And unlike Sanjay, that kind of prompted me, that made up my mind that I would always stay in academics and not go to industry. And my PhD also helped me decide that, viruses are great, but we are coming after the fact. So I want to find out things before anybody else finds them. So I want to go to the basics of biology, and that's what you will see we are doing cell division. What can be more basic than that? And so my postdocs, I also again took a break and, then joined CDFT. So even though I've highlighted these two, these 2 years as great, in Bangalore I had learned how to set up a lab, and the attitude that I can do anything, because that was the projection we were supposed to give to our clients. And of course networking, I just made a platform and told everybody in Bangalore that the next big thing in science is arriving. Just wait a few years. Of course then I went to Hyderabad. And then here, again I took a break, and the reason I'm highlighting this is because science is tough and everything will not be perfect. But the most important thing is to persevere. And it's all about you and your science and your motivation. This is not a race. So just continue and make sure that you end up at the table. It doesn't matter, if your batchmate is ahead, or if you're ahead, or if you're several years late. Just reach there. And just enjoy what you're doing. So that's the take-home message I want to give from my talk, and then we'll talk about other matters right now. So, as I said, that we've been interested in cell cycle regulation, and what we've been looking at is that cell cycle is essentially a set of genes getting activated and a set of genes getting repressed. And that happens, and that's how the phases come into being. And so we chose our transcription factor early on, which was E2F, and we decided that the most activating histone methylation, which is HTK4 methylation, is the one that we would be studying. And then we started focusing on the writers, which was the KMT2 family, and the erasers, which was the KDM5 family. So they deposit the mark and then they erase the mark. And what exactly are they doing here? So basically the idea was that people said that histone 3 lysine 4 mark was not erased during cell cycle, and it was only during differentiation that this happened. And we thought that that's not possible because genes are upregulated and genes are downregulated. So I think that was the first thing we showed, was that the eraser was brought to these E2F transcription factors, which by the way regulate a number of genes, including those of DNA synthesis and also in mitosis. So we said, okay, they regulate it and they erase this mark, and we were able to show that experimentally. And then the idea was that this mark is set every cycle, and in the G1S, the writers come in and then they deposit this mark and activate S-phase genes. And then these are repressed. So that was kind of, I think this was among the first reports to show that this mark is dynamic even during cell cycle. The other report was talking about circadian rhythm, and then most others talked about what happened during differentiation. So after that, we've been kind of interested in if they have other roles in cell cycle besides gene expression, or even if it's gene expression, what are these roles? So today I'm going to be talking about the HMT family, and there are 6 members here in humans. And the reason they've been put together is because of this set domain, which actually does the methylation. And then they have a couple of other domains which help them in chromatin recruitment. So these are also readers of the same mark, or they may read other marks. But what is most important is, so as you would see, the set 1A doesn't have really any reading domains. But MLL 1 and 2, which resemble Drosophila trithorax, and then 3 and 4 also closely resemble the Drosophila version, whereas these guys resemble the yeast. So they don't themselves work alone, but they actually require a couple of proteins which enhance their catalytic activity. And then they have content-specific proteins which would take them to specific gene promoters. So out of these, MLL is the founding member because of its role in leukemia. And I'll tell you just in a second. What is also interesting is it gets cleaved into 2 parts. The N-terminal and the C-terminal after it's translated. And there are 2 domains in it, unlike its other sisters. So this is the set domain, and it also has a transcriptional activation domain. And what's interesting is that maybe only 500 loci are methylated by MLL, but about 5,000 genes are regulated by MLL. So most likely it's the work of this guy. And then what also happens is that it is here at the genetic level that it translocates and fuses to random partners, and that's where it gives rise to a kinetic protein which is thought to give rise to leukemia. So MLL rearranged leukemia has diverse causes but the same result. And this is how MLL would be regulating. It occurs in a large complex where there's PAF and superelongation complex along with POL2. And it's thought that the MLL fusion proteins, they hijack this machinery and they give rise to illegitimate transcription, which is thought to cause leukemia. But what is interesting now is over 200 partners of MLL fusion are known, and only select few are involved in transcription. So from the time we started our lab, we have been advocating that there are other functions of MLL besides transcription which have to explain this. Transcription cannot explain all the causes of leukemia. So just to tell you what exactly the lab has been doing, so these days we are very, very interested in the non-coding region, just like Stefano has given you a base. So we are particularly interested in centromere and RDNA. And both of these have been difficult because of the repeat region and also because they are not annotated. So this year centromere was annotated, but RDNA is still masked in the genome assembly. So it's kind of difficult to analyze. And then of course we are also interested in what the fusion proteins do to this. So this is about the transcriptional role. But we've also been interested in how MLL or these enzymes may have a non-transcriptional role. And this is where we differ from the world. So they don't believe us, but now probably they've started doing it. And we say that they have other roles at spindle and centrosome. So spindle we've already shown and in the process of showing more, and centrosome is something that we kind of studied a lot and would be putting forth soon. So we are also studying how MLL works in rare diseases, so besides the leukemia. And this is the syndrome we've chosen, and the reason is because these individuals are haploinsufficient for MLL. And this is exactly the cellular model. So they are developing, they have, some functions are being redundantly compensated by the other sisters, some functions are not. So it will help us exactly find out what exactly does MLL do. And then of course the other part, the erasers also, we keep looking. So the idea now is that we are, trying to split, and I think we've achieved that. So don't worry, no one was harmed during the making of this slide, okay? So the question that, as I said, we wanted to know what exactly is it doing in the other phases of the cell cycle. So early on we found that once we removed MLL by siRNA, we found binucleated cells and they also had micronuclei. And this were indicative of defects in cytokinesis as well as segregation. And we found out that these functions were not dependent on the cell domain, but they required WDR5, which is actually, I showed you in the beginning that it was one of the components of catalyzing the methylation. So that was all interesting, and out of the other sisters, only SETD1A showed this phenotype. So it was unique to MLL and SET1A, which was very confusing because they are quite far apart. But that's what we observed. And then we started finding out that, in fact, once we removed MLL, we started seeing defects in mitosis. So this is where the metaphase plate, the chromosomes should be arranged, and this is the spindle. And once we removed MLL, they were all over the place, and spindle was either, there was more of it or there was nothing. And this was happening with WDR5 too. So then we found out that is it going to other places where it should not be, and we found it all over the spindle during mitosis. And people had reported before that it was also on the mitotic chromosomes. So to reconcile what's going on exactly, we took to live cells. And as you can see, this is the N-GFP tagged MLL. So it was majorly on the mitotic chromosomes, but it was also at the other places. So it was on the centrosome, it was in the central spindle, and then mid-body, etc. So besides this, we also found that MLL was actually on the centromere. So these small dots that you see, these are centromeres, or they would be called kinetochores in mitosis. So this is where we wanted to know if, so actually at this time, the centromere transcription was just coming up, and we thought that that's what we want to find out. We started early, we are arriving now, but I guess we still publish it in time. So this is, the centromere, this is how it looks like, two small dots on the chromosome. And even though, this is the base, which is CENPA, there are over 200 proteins which assemble on it during mitosis to form the kinetochore, which actually captures the spindle, and then the segregation takes place. So this assembly was getting hampered somehow, and that's what we wanted to see how. So as I said, that CENPA, which is actually nothing but a histone variant, and interestingly, even on the centromere, it is alternated by CENPA and histone 3. Okay? And what is also interesting is that the CENPA is deposited after mitosis in early G1. So that's also another interesting fact, because you would think that this should happen before mitosis, but it happens after mitosis. And one other fact that I want to bring to light is that we have heard of alpha satellite DNA, which we think occurs on all the centromeres. And we think all the centromeres have the same sequence. But that's not true anymore, and each centromere actually has its unique sequence, and then it has alpha satellite repeats also. So I'm going to be taking the example of chromosome 17, which has 3 arrays. So it has Z1, Z1b, and these are shown to form a centromere. So if this transcribes, then CENPA will come and deposit here. If this transcribes, CENPA will come and deposit here. The status of this guy is not known right now. So I would be using this again and again, and then these also, so they have been shown to carry the dimethylation mark on lysine 4, and also repressive marks. So this was very counterintuitive because these were thought to be inert region, transcriptionally silent, but that's not true anymore. So what's happening is, this is the model. So what people have said is that the dimethylation actually makes it open to transcription, and then Pol2 comes and transcribes, and the transcripts which are made, they actually prompt the AJURP, which is the CENPA chaperone, to deposit it during early G1. So this is how the cascade goes. And we wanted to know who's depositing this mark, or which MLL, because there's 6 of them, right? So that's where we started. And if the CENPA is not deposited, eventually the cell will start to missegregate its chromosomes. So we started by checking, what's happening, and here I just show you alpha satellite transcripts. So these are again non-coded, so we have to be very careful that you're not detecting DNA. And using the Pol2 inhibitors, they were going down. So one is the control. And what we also found is that all the MLLs were actually regulating it. So this is the ML levels going down upon siRNA, and this is the alpha satellite levels. So so we are showing you 5 here. 5 sisters at least were involved. And then we were able to see that yes, they were doing the chromosome 17 specific array also. And then this was via the centromere. So we made mutants, we complemented, and we were able to arrive at that conclusion. So next we wanted to see if these guys went there, and here I think this is very convincing that they are present at the centromeres during the mitosis, and then where the transcription takes place, and then deposition in early G1. And so even MLL2, 3 we could show, even though it's not as striking as these guys. So now before this, all the studies that I've shown you were taking place in human artificial chromosome. And we said we did not have access to it, and so we said we'll just go to the endogenous system. And even though this sounds like a regular chip, it took us a long time to standardize because the background would be enormous. So we would, because it's a repeat region of over 400 repeats, we would just keep getting high background. And so we took a lot of time to standardize it, and finally we were able to show convincing chip that yes, MLL and SET1A were binding, and these loci were dimethylated. And of course they were marked by other centromeric proteins which were not present on the other locus. So then we made MLL knockouts, which was another story, but we managed. And these were inducible knockouts, and here the MLL levels did go down. Okay. So after the MLL went down, we expected the dimethylation to go down. It went down. And then we looked for other activating marks. So like K9 acetylation, which is another activating mark, we saw it was going down. And then the K9 methylation mark, which is repressive, it was going up. So the centromere was closing up. But what was surprising was that the K36 dimethylation, which mirrors the dimethylation or the K4, this was going very high. And we were puzzled and we could not understand why this is happening. So what we started is, we started looking up at R loops, because there were reports coming in that the R loops are actually accumulating at the centromere and they are associated with K36. So R loops are nothing but RNA DNA hybrids, which are formed as a result of transcription or replication, etc. And sometimes they are promoting transcription, sometimes they are detrimental. So here in the IF, what we started with is, we started seeing, to our surprise, that when we knocked down MLL, the R loops went up. And in case of SET1A, they went down. So this was not what we expected, because both of them were depositing the mark, they should behave similarly. And once we inhibited transcription, they both went down. So the R loops were dependent on transcription, but both the sisters were behaving differently. And why? So we have, then we kind of, reproduce these findings by doing DRIP, which is, chip for DNA RNA hybrid. And we were able to show that indeed this is happening. Then in case of MLL, the R loops are high on the centromeres, whereas in case of SET1A, they are low. Okay. Now as to why it is happening, we have no clear answer as of today, because we have an MLL knockout, but we don't have a SET1A knockout. So in the MLL knockouts, we see that the Pol2 is accumulating, or the elongating Pol2 is accumulating at the centromere. So probably it's getting stalled and thus forming R loops. But because of the absence of SET1A knockout, we are trying to do these studies with the shRNA knockout. Now those of you who are familiar with chromatin protein would know that it's very difficult to knock them, or in the knockdown it's very difficult to see them go off the chromatin. Even if you don't see them on the blot, they end up on the chromatin. So that's where we are at right now. But then we went ahead to show that the CENPIA recruitment was actually hampered. So look at these panels here, and you would see that as soon as we remove the transcription activation domain or the SEP domain, the CENPIA was brought down. And this was surprising because people don't see this clear difference. They have to go to the nascent transcripts, which I'll show you in a minute. So here you block the old CENPIA by a quencher, and then you let it get incorporated, and then you see the newly labeled CENPIA, and that too was also going down in both MLL and SETD1A, and we verified this by the native CENPIA chip that indeed they were down on the centromere. So this is right now in revision, but what also while the study was going on, now this year centromere has been annotated on the genome. So immediately we went to chromosome 17 and we were able to confirm that MLL is coinciding with the dimethylation and the CENPIA. And what was also interesting is when you zoom out, there is more of dimethylation in MLL on the pericentromere. So that's something that we're going to look at and also see if it's present on all the centromeres of all the chromosomes or different ones. But this is kind of the end, and I think the summary is basically that yes, MLLs are depositing this mark and many of them are doing. We have just tried to show them with one. But what is also interesting is they have their differences because one of them causes RU accumulation but the other one does not. And we're trying to find the reason why. And I think this is the last slide where Kaushik here has actually done most of the work, and he was helped by Kaisar, Fire, and Rao. So, thank you.
Thank you, Vita. Very nice. Enjoyed the talk. I think we will have a tea break, 10 minutes, and we will assemble again. Oh. थोड़ा डिले उसले हुआ ना तो हम जल्दी शुरू कर देते हैं. It's okay. Let's begin with the next round of talks. Again, a star performer of ICGEB since his PhD days, Vivian Dasarathi. He was associated with malaria group, and but now he works on RNAi and various RNAi related process. So Das, please take this.
Thank you. And it's indeed nostalgic being here, and especially in this auditorium, where I think I defended my thesis. So yeah, so I just wanted to take 1 minute talking about my journey here. So my journey started in Jan 4, 2000, and I joined the malaria group for my PhD, and with Dr. Pawan Malhotra as my guide. And it's been a very inspirational and amazing journey for me with him. And I learned quite a bit. I learned quite a bit from him. And one thing I really learned is to not to give up, either on projects and people, and he never gave up on me. So I finished my PhD with him. And the two people who really inspired me, one is Dr. Sunil Mukherjee. He's not here. And he has been a tremendous mentor for me. And my passion to do RNA biology actually stems from his guidance, right? And another person is Asif, and he's there, right there, right? And man, I mean, I bothered him so much during my PhD time. He literally handheld me and took me through the thesis. And whatever molecular biology I learned is because of him. And thanks a lot to you. And of course, my journey would have been completely incomplete without the acknowledgement of other teachers and mentors throughout, right? And especially Dr. Chauhan, who was the director here, and he always insisted that nobody should do PhD more than 4 to 5 years. And that push actually made me complete my PhD in 2004, right? On that note, it would have been very, very disservice if I would have not acknowledged my friends. And that's my batch, right? And that's where my beautiful journey started with a bunch of people here, and some of them are there here. And then as I proceeded, I procured more friends, and my journey has been enthralling, enriching, and very fruitful. And the fruitful journey led to a fruitful outcome, that is that, right? So I got married to my batchmate, Yunita Raghavan. She did her postdoc from Yale, and she is head of RDO, Research and Development Office at NCBS in STEM, and she couldn't come today, and she apologized for that. On that note, I would like to go and talk about a little bit of my science here, right? So my lab is interested in understanding how stem cells are regulated, and how the internal and intrinsic and extrinsic factors regulate how stem cells behave, right? So one aspect we are looking at is extrinsic factors, and other factors are intrinsic factors. What my lab is actually studying is how cellular plasticity is controlled, and how that cellular plasticity actually helps in regenerating. And to study these processes, we use multiple model systems. One is Planaria, which is a fantastic regenerating model, which I will talk about in a minute. We also use mouse and human embryonic stem cells to understand cellular plasticity, and we also work at mouse early embryogenesis, because mouse early embryogenesis is controlled at the level of protein synthesis, not at the level of transcription, right? So, going back to the extrinsic factors, we look at one of the key extrinsic factor called extracellular matrix. Extracellular matrix is a mesh-like network in which tissues are embedded. And what we have shown recently is this. We have shown that the extracellular matrix acts as molecular highways on which the stem cells migrate and take specific lineage decisions. So maintaining the highway is important for the cars to go at a speed, right? In a similar fashion, maintaining the thickness of ECM is very important for the stem cells to migrate and differentiate into specific lineages. And we have shown that a transcription factor ETS1 is very critical to maintain the thickness, and the ETS1 maintains the thickness by controlling the expression of metalloproteases. Metalloproteases digest the ECM and thus control the thickness, and thus allows the stem cells to differentiate into a specific lineage. So that's the piece in the context of extrinsic factors, and we also look at intrinsic factors. And one of the intrinsic factors that we are looking at the protein landscapes, we are looking at the protein landscapes, and we are also looking at the metabolic states. Because by altering metabolic states, you can actually alter how the stem cells differentiate. And the changing metabolic states, also regulates how the protein content in that cell is changed. And one of the key factors that regulate the protein landscape is actually translation regulation. So what we have shown is this in translation regulation. So, and Stefano has beautifully put out about cat-dependent translation, and that happens by a 5' UTR interacting with the 3' UTR in a poly A tail. And there are proteins called as poly A binding proteins that actually initiate that translation process. One of the biggest bottlenecks in protein synthesis is translation initiation. And we have shown that by regulating the expression of a specific poly A binding proteins, you can actually regulate how the stem cell choices are happening, and that happens by regulating the protein synthesis. In addition to that, my lab is also looking at the length of a poly A tail. It's very interesting. If you take a cell, every mRNA has a different length of poly A tails. Just by altering the length of a poly A tail, you can alter the stability of the mRNA, thereby alter the protein synthesis and bring about lineage specifications. So we are looking at the mechanisms that regulate polyadenation here. And of course, my lab started looking at microRNAs, their binding to 3' UTRs, and how they destabilize mRNAs by binding to 3' UTR, thereby allowing the process of differentiation. So, why this is happening in the last 5 years, my lab got interested in this molecule called ribosome. The ribosome was thought to be a monolithic micromolecule which sits in the mRNA and does translation. So, but what we have shown in the last 5 years is this. We have shown that the ribosomes actively interact with the environment around. And we have done 3 different kinds of projects in that relation. We have shown that the microRNAs interact with a specialized class of small RNAs which are actually derived from tRNAs. The tRNAs break into 2 pieces, and a part of a tRNA broken piece goes and interacts with ribosomes and controls how ribosome sits on the mRNA. In addition to that, we are also looking at RNA helicases that regulate how the ribosome assembly happens, thereby you regulate translation. We are looking at specific regions on the rRNA which are called as rRNA expansion segments. And these rRNA expansion segments are critical for loading the ribosome onto the mRNA. And if you tweak the rRNA secondary structures, you tweak how the ribosomes are loaded, thereby you regulate the protein synthesis. And the last but not least is the post-transcriptional and post-translational modifications of the ribosome. You can modify the ribosomes at specific places, and then you can control how the ribosomes are being loaded onto the mRNA. So this is what is emerging from the lab in the last 5 years. This brings us to a point called as ribocentric view of regulation of translation, which is critical both for stem cell function as well as regeneration. Right? So what I will be talking about today in next 10 to 15 minutes is this. I will be talking about the metabolic states, and one specific organelle I will be talking about is mitochondria because it's a metabolic hub. And when I end the talk, I will show you how translation regulation can go back and regulate the metabolic states of mitochondria. That's what the talk is about. So this is a statement I am making. We know very well from the literature that if you change the metabolic states, you actually bring about stem cell differentiation into specific lineages, right? So this brings up to a very interesting Weddington model. This is a Weddington landscape where they have shown that the epigenetic changes are essential for stem cell fate choices. And the landscape they described is an epigenetic landscape. But this landscape is true for anything. This might well be a metabolic landscape. As the stem cells flow and take specific values, they take specific lineage decisions, right? So one metabolic state control of a cell is being mediated by this organelle called mitochondria, right? Because mitochondria is a hub for TCA, oxidative phosphorylation, and it is also a hub for oxidation, meat oxidation. So why we are interested in mitochondria? This is the reason. If you take a pluripotent stem cell, it is heavily glycolysis, right? ETC goes up and oxphos goes up and you lose that commutated version of the mitochondria. So it appears that you can actually use the mitochondrial states to define the pluripotency versus the differentiated state. The question that comes is, what regulates mitochondria? Can mitochondria be used as a functional organelle to distinguish the differentiated versus the pluripotent states? With that, we wanted to use a planaria as a model, and you will see why we are using a planaria model to address those questions. Why planaria? Planaria is a flat form. It lives in fresh waters, right? And it is freely available anywhere you look for in ponds. And the beauty is this, you can take planaria, you can cut planaria into small pieces, and each piece can regenerate back into a complete animal, right? And how it does that? It has a bag of stem cells distributed all over the body. And the red dots you see are the BRD incorporated cells, which are dividing cells here, which we call them as a neoblast. And these cells are actually has pluripotent properties that can allow them to regenerate, right? So a lot of work has been done in last decade or so, where they have identified markers for the neoblast, which are the stem cells of planaria. And one marker they have identified is called PV, right? But the PV is a global marker for all the stem cells in planaria. By looking at PV, you cannot say whether that neoblast is a pluripotent neoblast or a committed neoblast. How do we address this problem? So what has been done is this. They developed this fat sorting technique where stem cells are typically bigger in size, right? They have a dense nucleus, and the dividing cells has 2n plus DNA ratio, right? So they use that property here. So they took all the cells from the planaria, they strained with the DNA binding dye, and they strained with the cytoplasmic dye here, and they sorted 3 different populations of stem cells. What you see X1 is a highly dividing cell, and what you see in X2 are the cells that are in G1 phase, but the problem with X2 is it's extremely heterogeneous, and what you see X in cells is a terminally differentiated cell. So now what they did is, they excluded X2, they excluded differentiated cells, they took highly dividing cells, and they have done a lot of single cell transcriptome and transplantation experiment. And they have come up with this beautiful model, which we call as a hierarchical model. What does this model say? This model says this. You have a clonogenic neoblast, which we call them as a pluripotent neoblast. The pluripotent neoblast, upon perceiving signals, differentiate into committed neoblast, and the committed neoblast will differentiate into progenitors. And the committed neoblast and pluripotent neoblast are marked by this marker called PV, right? But the PV cannot distinguish between pluripotent versus committed. So they created this hierarchical model saying that there is a linear flow of stem cells here. But it has been thoroughly challenged in the recent paper where they said there is no hierarchy in terms of pluripotent versus committed. What they said is, when a stem cell enters this cell cycle, as they proceed through the cell cycle, they acquire differentiation marks and they undergo asymmetric division, and the side which has the differentiated marks will become a progenitor, and the side which doesn't have a differentiated mark will become a non-committed cell or a transitory pluripotent stem cell. So we are back to square one, still not understanding what is a pluripotent state in planaria, right? In order to understand the pluripotent state, we actually try to address that, which is actually the outline of my talk. Can the mitochondrial states distinguish pluripotent versus differentiated? Is the mitochondrial state changes a consequence of neoblast differentiation, whether it's a cause or a consequence? What is a metabolic state? If we can sort out the cells using mitochondrial state, can we define what is the metabolic state of a pluripotent neoblast? Then we went and find out what are the molecular regulators that regulate mitochondria, right? So let's go here. If you want to look at pluripotent state or identify a pluripotent state, you cannot look at the dividing cell, right? You only have to look at the cells that are in G1 phase. So what are the cells at G1 phase? We call them as X2 cells. But the problem with X2 cells is it is extremely heterogeneous. In order to rule out that heterogeneity, we took the X2 cells, we stained them with mitotracker. Mitotracker actually stains the mitochondria based on its potential and also mitochondrial content. And then we also used forward scatter. What does forward scatter do? I said the neoplasts are bigger in size, the stem cells are bigger in size, right? When you shoot a forward scatter, if the forward scatter is high, that says that the cell is big. So we use that property to actually identify different cell populations in the X2 here. And what we did is we identified 4 different populations here, which is X2, X2 MTG low, X2 MTG high, high forward scatter, low forward scatter, and for also MTG high, high forward scatter and low forward scatter. So these are the 4 populations we took. And we know that the PV is a stem cell marker, so we did RT-PCRs and we have shown that X2 MTG low, high forward scatter, that means the bigger cells have a high expression of PV, suggesting that they could be indeed a true stem cell populations. So this is just the summary of the work, which says that the X2 MTG low HFSCs are indeed the true stem cell populations of planaria, which are in the X2 population during the FACS sorting here, right? So how do you validate this? The beauty of planaria is transplantation experiments, right? So now, if I say these are pluripotent, I need to prove that. The way I can prove that is, you take planaria, put them on irradiation. When you radiate the planaria, you kill all the neoplasts. All the dividing cells will die. Now you take this cell type and you inject it into the planaria and ask the question, does it rescue planaria or not? And that's what we did. And we did this transplantation experiments and we almost saw around 80 to 90% rescue. That means when we took this HFSC cells, which are 2M MTG low HFSC cells, and we injected them into planaria, we could actually see an 80% rescue of planaria, suggesting that we indeed actually identified a pluripotent stem cells in the X2 populations are in the G1 phase of the cell cycle, right? And we can show here how much the rescue is. The rescue is way better than the other cell populations that were detected here. So, so what is next, right? The next is we have identified a pluripotent state in a G1 phase of the cell cycle. Now, and we are saying that they are low in mitochondria, and when they differentiate, they become high in mitochondrial content. The question we are asking is this a cause or a consequence, right? In order to address that, we just did a simple experiment. What we did is we blocked the mitochondrial activity and we wanted to see whether the pluripotency rate increases or not, right? So for that, what we did is this experiment. We took stem cells from planaria, we developed an in vitro system where you can actually put the stem cells of planaria on the in vitro system and maintain them for 48 hours. Beyond 48 hours, they just differentiate. So we took those cells and we treated that cells with FCCP, which is an inhibitor of ETC. But basically, it's a decoupler. When you do that, what we found is the differentiated populations vanish and we see an increase in this MED1 population. So now we have to show that this is pluripotent, right? So we took these cells and we injected the cells in planaria and we have seen that 100% of the planaria is rescued. Not only they are rescued, we also see that the stem cell populations are spreading, suggesting that this population is indeed a true pluripotent population that can be used further to characterize the metabolic state and also the regulatory dose, right? So that's the summary of the work that we have done so far. What we have shown is we have a pluripotent stem cell, and as the pluripotent stem cell differentiate, the OXPHOS goes up, right? And we have shown that if you inhibit the OXPHOS, you actually retain the pluripotency for a longer time in planaria. And this could be a beautiful way actually to identify the true pluripotent stem cells in any animal system. The next question we ask is this. Since we know what are the pluripotent stem cells, we can identify the markers that are enriching that. In addition, we can also define the metabolic states, right? So in order to do that, what we did is we took this 4 population, and this is our pluripotent population, and we did single cell transcriptome. I'm just summarizing the result here. I'm not going into the depth of this result. This is a summary of the single cell analysis that we did. We identified X2 MTG low HFSs, which is this population, has a highest expression of stem cell marker. which validates our previous finding that these are pluripotent stem cells. And when we took this from the single cell transcriptome, we asked how many clusters of cells are there in this population, and we found that there are 15 different clusters in that population, right? And then we asked, among the 15 different clusters, which cluster is actually a pluripotent cluster, and we found that the cluster 7 here is a pluripotent cluster. And now, to summarize this result, we started with 20,000 cells by doing single cell transcriptome. By using different layers, we could actually identify 149 cells that exhibit the pluripotent markers and that could actually rescue the planaria. We took that 149 cells and asked what are the genes that are specifically expressed in 149 cells, and we identified 256 genes that are exclusively expressed in this population here. So we went ahead and we did, of course, Go annotations are the easiest to do. We did that and we could identify a metabolic pathway. And this metabolic pathway genes are very interesting. What we found is this. We found that in this population there is a very high glycolysis, and what we found is the glycolysis doesn't run towards the pyruvate. The fructose bisphosphate, which is an intermediate, is shunted to pentose phosphate pathway to make ribosugars because these are actively dividing cells, right? If you don't run it through pyruvate, where is the energy source? In order to have an energy, what the stem cells are doing is they overexpress lipid transporters. They take the lipids from the intestine and they break it down by beta oxidation. So we see an increase in the beta oxidation in expression. And what we interestingly saw is the TCA also runs partial. The TCA cycle, if you remember the TCA cycle, only runs to succinate. It makes a lot of alpha ketoglutarate and citrate. And alpha ketoglutarate is a cofactor for methylation and demethylases. So that's the reason why it runs off TCA. It runs beta oxidation for energy, and it uses glycolysis to actually generate ribosugars. So by using single cell transcriptome, we actually dissected the metabolic state of this pluripotent stem cell, and not only that, just 1 minute, I'm done. Just 1 minute. Not only that, we also identified a lot of translational regulators. This is where I said I will come back and link it with protein synthesis, right? We identified RNA helicases. We identified an interesting set of protein called as Tudor domain proteins. These Tudor domain proteins are essential for RNP granule formation, and we also identified proteins that are essential for mRNA stability, right? So we are interested in Tudor domain protein because if you take a pluripotent stem cell, there are a lot of RNP granules in the pluripotent stem cells. That made us interested in the Tudor domain protein, and this is the nutshell of Tudor domain protein work that we have done. We have shown that they are part of RNP granule, they are in very close vicinity to mitochondria, and the RNP granules are actually, when you pull down the TDR-D9, which is a Tudor domain protein, you actually pull down mitochondrial transcripts, right? So this is the working model that we have. I will just skip this part. This is the working model. You have a transitory pluripotent stem cell, and the mitochondria is inactive. And you have RNP granules floating in the cytoplasm near the vicinity of mitochondria. And what we found is these RNP granules actually carry the transcripts that are essential for the activity of mitochondria. So RNP granules are actively involved in repressing the protein synthesis, which makes proteins that are critical for mitochondrial activity. 95% of the transcripts for mitochondria are actually made in the nucleus. They are transported to the cytoplasm. In the cytoplasm, they are translationally repressed and kept mitochondria inactive. When you have a differentiation signal, the TDR-D9 is dissolved, the transcripts are released, they undergo protein synthesis, and the proteins are made and translocated into the mitochondria, and thus mitochondrial activity goes up. So we actually went ahead and identified the factors or molecular pathways that regulate the mitochondrial activity in planaria. So we started with this confused state of what is pluripotency. As we proceeded further, now we know what are pluripotent stem cells. It's not just we know what are pluripotent stem cells, we also know what the metabolic states are, and we know what controls mitochondria. So that's where we are, and that's the group. I mean, most of them left in my group don't think my group is this big.
there is a shift in the program. Manish, who has to go somewhere, he will present first, and then Amit Singh, who is the boss here in ICGEB for a long time, 2-3 years. Yeah. And then we can have lunch, and after lunch we can have a talk from our guest from Vietnam. Okay.
So very good morning, everyone. I guess that's another nostalgic start. It's good to
a while. It's great to see all friends and colleagues here. And ICGEB did really played a very important role in my career because I started my research program here, and because of the foundation here, been able to establish and carry forward the same intensity of research program at ISC also. So thank you all, and really appreciate all the collaborations and students' effort. So today I'll be talking about mycobacterium tuberculosis, and the title gets very complicated, so don't worry about the title. All we want to do is that we want to somehow increase the efficacy of current anti-TB drugs, whatever are there in the market. And we want to do it rationally. We want to look into the mechanism and then try to solve complex problems with simple tools. So that's what the motto of the lab is. So if you look at tuberculosis, it's a very recent statistical analysis, and it tells you that TB is an ever-growing problem, and about 2/3 of tuberculosis cases are actually contributed by 8 countries, and India being at the top. So this is what the scenario is, and why it is really a complicated problem. Why tuberculosis management is difficult is because it's a disease which sort of presents a spectrum. And that spectrum ranges from latent tuberculosis infection to active cavity diseases. We have been for years studying about latent TB and active disease. Latent TB is a disease where probably bacteria are there in your system, but there are no clinical signs or symptoms. And active cavity disease is actually where bacteria are multiplying rapidly. Cavities are there in the lungs, and they are able to transmit infection. Now, over the year, it has been clear that there are different forms of tuberculosis. While latent and active TB are easily recognized, these tuberculosis cases which are sort of progressing from latent to active, incipient progressing disease, are difficult to track and therefore difficult to treat also. And if there is some kind of biomarker which help us telling that some of these latent tuberculosis population can eventually progress into active TB cases, we can start treatment earlier, and that can halt transmission of this disease. And second is active uncomplicated form of disease, which is non-cavitary active TB. This can be detected by simple X-ray and sputum test. You need to differentiate between active uncomplicated form, where there are no cavities in the lung, but bacteria are replicating, and cavitary form. And recent clinical trials sort of indicate that if you can, these patients which doesn't have cavitary disease, they can be treated with 4 months of anti-tuberculosis drug. While it becomes cavitary, you need to treat these patients for 6 to 9 months. So there are really interesting discoveries now going on which are able to cut down treatment time. Conventional long-term treatment is almost for non-complicated cases is for 6 months, and there's been a really bigger problem because 6 months is too long treatment time for 4 drugs, and there is always non-compliance. Compliance is a real problem leading to development of drug resistance. So we need short therapy time. We need short course therapy. Recently, clinical trials with rifampicin and moxifloxacin seems to suggest that you can reduce therapy time to 4 months, and approval is pending. And we need really long. We need therapies which can reduce therapy time so that emergence of drug resistance can be curtailed. Looking further, I want to tell you why really we need, what is a problem in a sense, is that you will know that when TB is in different tuberculosis lesions, you will see that there are different microenvironment. In vitro culture TB in test tube is very simple. We always study it in our DSL3. It is actively metabolizing, there is accelerated replication. And drug sensitivity is also quite high. Once you move from this stage to macrophages and to lymph nodes and to cases granulomas, things become complicated. This form of granuloma is more evident in active TB patients where you have vasculature, blood flow is there, bacteria is replicating, and there is more lesional permeability of drugs also, so it can be targeted. If you look at cases granulomas which are more evident in latent form of tuberculosis, here bacteria is extracellular, it's in cases environment. There is avasculature, oxygen supply is these persist or reactivates and cause infectious disease. So as you can see that, from active metabolism, accelerated respiration to slow metabolism, deaccelerated respiration is coupled with reduced killing by drugs. So one of the approach of our lab is to understand what are the signals which MTB is sensing in this environment, and then allow them to switch from active metabolism to slow metabolism and adopt this drug tolerant state. So work with the signal. And secondly, if there's a way wherein we can reverse the slow metabolic state to an active metabolic state where bacteria start to accelerate respiration, and then we can sort of switch from drug tolerance to drug sensitivity. So what we recently published is that acidic pH of macrophages seems to play a very important role in inducing drug tolerance and mycobacterium tuberculosis. If there is high acidity, bacteria become drug tolerant, and using that information we have been able to come up with a anti-malarial drug chloroquine which deacidifies macrophages, does not allow bacteria to become drug tolerant, and that just by adding chloroquine with anti-TB drugs we were able to successfully eliminate tuberculosis from mice. So those studies are going on. In today's talk, we have adopted an approach, and I'm going to show you some data where we were able to reverse slow metabolism or deaccelerated respiration to active metabolism, and therefore we were able to potentiate the action of a anti-TB drug, moxifloxacin, which is used to treat MDR patients. Now, so moxifloxacin is a very interesting molecule, and the reason is that it's very, very effective in murine model of tuberculosis. If you combine moxifloxacin with anti-TB drugs like isoniazid and rifampicin, it's able to clear infection faster in murine model. But if you start doing this experiment in patients, you don't get the data. In patients, moxifloxacin is unable to reduce therapy time. Therefore, there is something different in human tuberculosis lungs as compared to mice. And it became very clear with multi-mass imaging that moxifloxacin is actually not reaching in the central cavity caseous region of granuloma. So where these non-replicating persisters are residing, you don't see moxifloxacin reaching in those areas. And therefore, it appears that moxifloxacin would be suboptimally permeating in the diseased lungs and cases granuloma, and therefore it is ineffective in clearing infection. How does moxifloxacin work? Now, there is a lot of work done in other bacteria like E. coli and staph, and it becomes the mechanism becomes clear. Moxifloxacin is a fluoroquinone. For the students, it actually binds to an enzyme called as DNA gyrase or topoisomerase that removes superhelicity in the DNA when DNA is replicating. So the way it acts is that it binds to DNA gyrase when it is already bound to DNA duplex. So there's a ternary complex formation between the drug, DNA gyrase, and duplex DNA, and it tricks this enzyme to cleave DNA, but does not allow it to reseed. So ligation is sort of inhibited, and therefore there are a lot of DNA cut across the genome, and that's how it stalls DNA replication and kills bacterial growth. And there seems to be two mechanisms working here. One is that if your DNA repair machinery is working nicely, then it appears that the killing mechanism of this drug is dependent on protein biosynthesis and also formation of reactive oxygen species. So if reactive oxygen species are not formed or protein biosynthesis is stalled, moxifloxacin is not going to work if your DNA repair machinery is working. If there are no DNA repair machinery, if DNA repair is ineffective, then your cells are unable to repair damaged DNA, and killing happens in a ROS or reactive oxygen species independent manner. So this is what we need. We know from other bacterial systems. We don't know this information in Mycobacterium tuberculosis. So we think that if we are able to generate the same information to figure out if reactive oxygen species is regulating utility of moxifloxacin in MTB, like it does in E. coli and staph, there is a way to potentiate the activity of moxifloxacin in the case of tuberculosis. And we wanted to understand what is the lethal mechanism, and if we can understand the mechanism, we can also impact development of drug resistance. So somehow we want to improve the lethality of moxifloxacin even when it is reaching at a suboptimal concentration in the lesions. mechanisms that potentiate the action of fluoroquinol. So we did this experiment, and this is a biosensor which actually we developed in ICGEB, and still we send these plasmid across to a lot of investigators through ICGEB by signing MPAs and all. And it's been developed by a fantastic postdoc, Ashna Master. She's still working in ICGEB. She's back in ICGEB, I think. So she may be there in audience. And with the help of Dheeraj. So this biosensor is genetically encoded, redox-sensitive GFK, which we can express in Mycobacterium tuberculosis. And just by measuring the ratiometric fluorescence of biosensor, you'll be able to figure it out if bacteria is exposed to oxidative environment or reductive environment. So if there is an increase in ratio, bacteria is experiencing oxidative stress. If there is decrease, it's a reductive stress. So we express this biosensor in MTB and expose MTB to moxifloxacin, and you can see that in a time-dependent, in a concentration-dependent manner, there is increase in biosensor ratio, which indicates that MTB is experiencing oxidative stress in response to moxifloxacin, which is very, very consistent with E. coli and staph aureus. So there is nothing new in it, and it actually it is proportionate to killing also. So oxidative stress is being generated by MTB in response to moxifloxacin. We did RNA-seq, and we sort of found that, as expected, you would see DNA repair is going up in response to moxifloxacin because it targets DNA, is damaged DNA, so you are expecting to see DNA repair. Antioxidant machinery is also going up in response to oxidative stress generated by the antibiotic. What is interesting to us is we saw that energy and respiration is actually going down. And this is something which is in contrast to studies done in E. coli and staph, where in response to fluoroquinol antibiotics, you see energy and respiration is going up. Because hyperactivation of respiration is likely to produce reactive oxygen species. Therefore, it is very surprising to us that while the MTB is able to produce reactive oxygen species, it is unable to do so by increasing respiration. So that was something which was new in case of tuberculosis, and tuberculosis may not operate in response to fluoroquinol as E. coli and staph. So we, based on the transcriptomic data, we also measured respiratory potential of MTB in response to moxifloxacin using Seahorse bioenergetic analyzer, which non-invasively tracked how much oxygen is consumed by MTB under any stress condition, and also measured glycolytic rate. So we checked both, and we figured it out that in response to moxifloxacin, if you look at untreated MTB, which is not stress, there is increase in oxygen consumption, which tells you that active respiration is going on. If you expose MTB to moxifloxacin, you see there is de-acceleration of respiration as compared to untreated, and also glycolytic rate goes down, which is measured as a function of extracellular acidification due to production of lactate from glycolysis. So under untreated condition, you see a spike in glycolysis, then there is downward trend, but with moxifloxacin treated MTB, you don't see any increase in glycolysis, it just goes down. So it appears that, and what MTB is trying to do is that to take care of toxicity due to moxifloxacin, it is sort of slowing down its respiration and metabolism so that it does not get killed by this anti-TB drug. We also figured it out that if that is happening, it is expected that you will see accumulation of NADH, because if respiration is not happening, you won't see reoxidation of NADH to NAD. NADH is the electron donor that donate electron to respiration, so you won't see that, and therefore you will see accumulation of NADH. So in response to moxifloxacin, we do see accumulation of NADH, and that sort of gave us a hint that why we see increased reactive oxygen species production, despite the fact that respiration is sort of stalled in response to moxifloxacin, because the fact that NADH is able to generate reactive oxygen species by two mechanisms. A, it is able to release or leaches out iron from iron storehouse like ferritin, and it also reduces oxidized form of iron to Fe2+. Fe2+ reacts with hydrogen peroxide to give you hydroxyl radical, which is one of the major reactive oxygen species that damages DNA. So NADH accumulation in a way tells us that probably we see increased oxidative stress because of NADH mediated fueling of penton reaction. And this having this information in our hand, we sort of gave this model that moxifloxacin-mediated respiration arrest probably reduces its efficacy because you see, respiration is not going up, so you would see lesser oxidative stress, but we do see increase in raw surge due to accumulation of NADH. That may not be optimal, but still we feel that moxifloxacin activity can be counteracted if we are able to reverse this mechanism. In other way, if we are able to sort of reverse de-accelerated of respiration in response to moxifloxacin, we may be able to produce more reactive oxygen species here and may be able to potentiate the activity of moxifloxacin. So how to do that, that is the second question, and that is where we sort of stumbled on N-acetylcysteine, and Manisha alluded a bit in previous talk. N-acetylcysteine is a very interesting molecule because it is known, it's a very non-toxic drug used in many clinical conditions, in many respiratory tract infections, including tuberculosis, but it has been used to reduce hepatotoxicity and drug-related toxicity. It is known to scavenge oxygen, and it is also known to replenish glutathione. However, in case of tuberculosis, it has been shown that it also increases respiration. The mechanism seems to be not known, but it is known that if you use cysteine or N-acetylcysteine, somehow tuberculosis stimulates its respiration potential. So we did this experiment, and we thought, why don't we use N-acetylcysteine to potentiate respiration? If we are able to reverse de-acceleration of respiration, which is going on with moxifloxacin, we may be able to potentiate the activity of moxi. So we did biosensor response first with N-acetylcysteine to see if N-acetylcysteine is able to induce oxidative stress in NTB in response to moxifloxacin. So you will see that moxifloxacin is able to induce oxidative stress, but if you combine moxifloxacin with N-acetylcysteine, the amount of oxidative stress is higher. We sort of indicate that perhaps respiration stimulation is happening, and also survival also gets, survival also reduced in response to moxifloxacin plus NAC combination as compared to moxifloxacin alone. So by simple small molecule donor of cysteine, we are able to increase oxidative stress in MTB in response to moxifloxacin, and also potentiate the killing activity of moxifloxacin. This 3 hours experiment we did to see if NAC is indeed stimulating respiration in TB. So if you take wild type TB, this is how respiration potential looks. If you expose tuberculosis to N-acetylcysteine, you will see increase in respiration. And if you do the experiment with moxifloxacin doesn't show increase in respiration, but if you combine moxi plus NAC, you see respiration is stimulated. So we are able to stimulate not only respiration, but also oxidative stress by combining moxifloxacin with N-acetylcysteine. Now the question comes, is it potentiating killing in animals? Because that's the main thing. Before we can think of any sort of clinical trial. So we, as a proof of concept, we did 2 experiments here. First experiment is that we infected mice with tuberculosis for 21 days. And then after that, we did 10 days of treatment with the N-acetylcysteine alone, moxifloxacin, and moxifloxacin plus N-acetylcysteine. And you can see this in this figure that N-acetylcysteine, for only 10 days of treatment, this is a short-term experiment, this kind of pilot experiment, we were able to potentiate killing in the lungs a bactericidal antibiotic, moxifloxacin. So this was the last slide. So this is a summary model which we have provided here, where it appears that moxifloxacin, by making a primary complex with DNA gyrase and DNA, sort of inhibit replication, alter transcription, that leads to deacceleration of respiration, increase NADS to NAD balance, which leads to increased pool of mobile iron and increased loss. However, if you combine NS system, you are stimulating respiration, that gives you more reactive oxygen species and probably more than. So this was the last slide, and this is my group, and I acknowledge all the funding agency and collaborator for this supporting this study. Thank you very much. So I don't know about the mitochondrial. It doesn't. It is very specific. To bacterial DNA gyrase.
Yeah.
It doesn't affect mitochondrial.
Yeah.
DNA or topoisomerase.
So what is the link between transcriptional deregulation happening because of that and mitochondrial blockage in the the inhibition of respiratory chain?
The only there appears to be two ways this thing is working. First is the primary complex which is formed between DNA gyrase, that stores DNA replication, because there is a complex sitting between drug, DNA gyrase and DNA is clean. So there is no way replication fog is going to be increased. Now this somehow sense-wise probably transcription factor we still don't know, which leads to this transcription alteration. The immediate effect is vector stasis, and because it doesn't kill immediately. For killing to happen, that turning complex has to be released from the replication frog, so the DNA damage is exposed. Right? As long as it is sitting, it is not going to kill, but it is going to inhibit bacterial growth, and that inhibition gives a lot of time for antibody to realign its transcription and adapt in response to moxifloxacin. As a part of realignment, there is deacceleration of respiration.
Hi Amit, a great talk. So this moxifloxacin, you have already partially answered that this moxifloxacin is not for the vertebrate cells, the vertebrate gyrases, but I would imagine that there are other gyrase inhibitors, chemotherapeutic agents, which are already being used in cancer. So would you imagine that this N-acetyl lysine would have a similar global effect or similar effect also in other vertebrate system where it would potentiate the death?
Yeah, so N-acetyl seems to be a very interesting molecule in the context of mammalian system. It generally serves the purpose of, it has 2-3 different ways. One is that it is mainly as a source of glutathione. So if your cancer cells are experiencing oxidative stress, by putting N-acetyl you can reverse it just by restoring glutathione levels. It also can scavenge radioactive oxygen species directly, but that kinetics of that reaction to happen is very low. Very low kinetics. And third possibility is that it also releases hydrogen sulfide gas, which Manish was alluding to. And hydrogen sulfide, being gastrotransmitter, can do multiple functions, including reducing inflammation and taking care of, maybe it has anti-cancerous properties also. So there are a lot of possibilities. It's not.
So what following up?
The process seems like it is stimulating respiration.
Sure. Thank you.
Amit. Yeah, Amit, just curious, like just giving N-acetylcysteine, the MTB is developing resistance to moxifloxacin. So that I couldn't correlate.
Yeah,
right?
so no, it's not developing, it's reducing resistance. So moxifloxacin resistance is actually accumulating if you expose infected animals with moxifloxacin. Just by giving moxifloxacin, because of adaptive mechanisms, bacteria is able to come out of lethality and develop resistance. What we are saying is that one of the mechanisms to subvert resistance is that if you are able to use N-acetylcysteine, because it can induce more ROS and it can accelerate more respiration, kinetically you are going to kill bacteria faster and does not allow time it takes to develop resistance. So it's just a kinetic effect which N-acetylcysteine is having.
Thanks, Amit. Very nice talk.
line 5. Okay, thank you.